All right, everybody, welcome back. And in this video, we are going to be covering learning goals 4 through 9. And so part 1 was covering the first 3 learning goals, which was essentially muscle anatomy. In learning goals 4 through 9, we'll be covering muscle cell physiology. And then 10 through 12, we'll be covering whole muscle physiology. So let's go ahead and get started. So we're going to begin this discussion by talking about the sliding filament theory. This theory or model states that during contraction, our thin filaments are going to slide past our thick filaments, thus causing actin and myosin to overlap more. It's important to note that neither the thick nor the thin filaments are actually changing their length. They're just going to be overlapping more. And so what I mean by this is if you look at this section here, the thin filaments noted in blue and the thick filaments noted in this peach color are partially overlapping. And this is a relaxed state, whereas in this section down here, the bottom, the contracted state, you can see that the blue lines have essentially just moved towards this M line even more. And it's important to understand and sort of pay attention to the fact that the length of the thick filament is still the same on both positions, in both top and bottom, in both relaxed and contracted. And really the same goes for our thin filament. So if we start here at the Z line, and I highlighted one of the thin filaments, if we compare that to the length of the thin filament in this contracted phase, they're the exact same length. The only difference being that in the top picture, they weren't fully overlapped, whereas in the bottom picture, they are completely overlapped. So let's talk a little bit more about this sliding filament model. So as I just mentioned, whenever the muscle is relaxed, the thin and the thick filaments are only slightly overlapped, just at the ends of the eye bands. However, during contraction, these things called cross bridges are going to start happening, and these are going to generate forces that will essentially move the thin filaments closer to the M line. Okay, and so just a reminder, here's our M line right here in the middle of our H zone. And because these cross bridges are occurring, these thin filaments are essentially just moving towards the M line. Okay, so we're just moving this way. All right, and because of that, the whole cell becomes shorter. All right, so kind of going off of that, cross-bridge attachments, they're going to form and they're going to break several times. And each time they're formed and broken, the thin filaments are going to move closer and closer towards the center of the sarcomere. So this is going to be sort of a ratcheting type action. And overall, as you can see, this would cause shortening of the muscle fiber. And so I just want to point out, I haven't paid much attention to the next thing that I'm going to draw up here for you, but here is a relaxed muscle cell there on the top. And here we can see a contracted muscle cell. So as you can see overall, this process is going to cause shortening of the muscle cell to a contracted state. As it's doing this, there are certain things that we should keep in mind, including what's happening in all of our zones and lines and so on and so forth. So first and foremost, the Z lines, which you can see there, and here's another one over here, they are going to be pulled closer towards the M line. And remember, the M line are these set of 3 lines, which are just proteins right here at the very center of the H zone. Okay, so the Z lines are coming towards the M line. Additionally, the length of our eye bands are going to become shorter. And so here's a reminder, here is the length of one eye band during the relaxed state. But during the contracted state, you can see the total length here in the contracted state has become shorter. Additionally, because the thin filaments are moving towards this M line here, this causes the Z discs to become closer to one another, right? So if we're looking at total length here, we have Z disc to Z disc in the relaxed state, Z disc to Z disc in the contracted state, and you can see that they are much closer to one another in the contracted state. Additionally, the H zones are going to disappear. And as a reminder, the H zone is this entire area. As you can see from the bottom picture down here, now we have thin filaments that are penetrating that H zone, thus leading us to say that there is no more H zone. Okay, so all these blue lines are now in what used to be the H zone. Thus, at this point, we no longer have an H zone. Okay. All right, so I'm going to just play a quick video for you, and hopefully this can give you sort of an animated idea of what exactly is going on.
We move our bodies by contracting skeletal muscle. While the results of this action are plain to see, the mechanism behind the action is not. Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached at the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
All right, so I just think that is a really great video showing you in a real-time sort of situation what would be happening in that sliding filament model. Okay, so in this next part, I'm going to just start briefly describing some general steps that are taken to skeletal muscle contraction, and then we'll go into the more specifics. So the decision to move is activated by our brains, and the signal that our brains send is transmitted down our spinal cord to activate motor neurons, which then go on to activate muscle fibers. So neurons and muscles are muscle cells. They're excitable cells that are capable of what are called action potentials. And action potentials cross from the neuron to the muscle cell whenever acetylcholine is present. All right, and so this is just a really brief sort of introduction, and we're going to get into the details, so I won't spend too much time on them here. So this is just a quick little intro that I'm going to do for you. All right, so ion channels play a huge role in the ability of our muscle tissue to respond to a neuron signal. And in general, we have two different classes of ion channels. First are the chemically gated ion channels, and that is what you see up here. And the chemically gated ion channels are opened by chemical messengers, such as neurotransmitters, and in the muscle cells, it is acetylcholine. And so originally, before acetylcholine is there, what we have is a similar sort of setup. The only difference being that it's blocked off at some point. Okay, and so this channel is not actually open. We're just going to put those in there and pretend like it's blocked off. Now what happens is when acetylcholine comes in, acetylcholine is going to come in and it's going to bind to the receptor like you see here. And when it does that, that allows those little blockages to open up, which is what you see happening over here. And this is going to allow the movement of different ions on either inside or outside of the cell. All right, so when you're looking at these pictures, essentially what you're looking at is Let me just get my little pin out for you. Out this part up here is outside of the cell. This section would be the cell membrane, and this would be inside of a muscle cell down here. And so whenever these channels open, certain ions or certain charges are going to be able to enter the cell, and then certain charges that are inside of the cell are going to be able to exit. Okay? Alternatively, voltage-gated channels, voltage-gated ion channels, open or close in response to a voltage change in the membrane potential. And membrane potential is generated because there are different amounts of positive and or negative charges inside and outside of the cells. Okay, and so for example, say outside of the cell, we have 4 positive charges. And then let's say inside of the cell, we have 2 negative charges. Because there are different amounts of positive and negative charges inside and outside of the cell, this creates basically a difference. It doesn't add out to be 0. And because of this difference, energy, and typically in this case in the form of voltage, can flow, thus creating a net result for electrical current to pass through. And we'll talk about this more as we move on. Okay, so I mentioned a couple of slides ago, but again, skeletal muscles are stimulated by motor neurons that come from the somatic motor side of the nervous system. So essentially what's happening here is we have the brain up here and the spinal cord are going to give off neurons or nerves in this case, and we're going to say that this is a nerve, and nerves tend to have both somatic, so this would be both motor and sensory, but we're going to focus specifically on motor because motor leads to movement. And so I just want to highlight some important points here. The axon is going to travel down and it's going to end on a muscle. And in this case, we're going to say that it's the biceps muscle. And you can see at the end of this how it branched. It kind of did this sort of thing. You can't really see it very well over there. So it sort of did that kind of thing right there at the end. And each of these little things are called the end branches at the axon terminals. So these are little axon terminals that are located on the end branches of the axon of a neuron. These branches are going to end up contacting muscle fibers. And it is the contact between these two regions here. So between the nerve and between the muscle fiber, that's going to make up what's called a neuromuscular junction, or also sometimes called a motor end plate. And so here is one neuromuscular junction. We have another neuromuscular junction. Here's a third neuromuscular junction. And so each muscle fiber is going to have one neuromuscular junction that contains one motor neuron. All right, so this is just reiterating kind of what I was just talking about, but let's break it down a little bit even more. So the axon terminal, which is basically the end of the axon, and the muscle fiber are separated by this gel-filled space that you see in gray here, and this space is called the synaptic cleft. On the neuron side of things, we have synaptic vesicles located just at the end of these neurons. And inside of these synaptic vesicles, we have acetylcholine, which is a neurotransmitter. Okay. Additionally, on the muscle side of things, we have these infoldings of the muscle. And these infoldings of the sarcolemma are called junctional folds. And these junctional folds contain receptors for acetylcholine. Okay. And so the neuromuscular junction as a whole consists of axon terminals, the synaptic cleft, and junctional folds. Okay, that is the neuromuscular junction. So over the next few slides, we're going to talk about each of these 4 different steps, but here's the big picture. In order for skeletal muscle to contract, these 4 things have to happen. We have some events that are going to take place at the neuromuscular junction. Muscle fibers have to be excited. Excitation contraction coupling has to occur, and cross-bridge cycling has to occur. And all 4 of these events will lead to our skeletal muscles contracting. And so let's start going through each of these things individually. So first we'll talk about the neuromuscular junction. This is what happens at the neuromuscular junction. An action potential is going to arrive at the axon terminal. And then at the axon terminal, so we're seeing this little axon terminal here synapsing on a muscle fiber. So we're just going to zoom in on that so we can see exactly what's happening. So after the action potential arrives at the neuromuscular junction, the axon terminal, this change in voltage caused by the action potential opens up calcium channels. And these calcium channels are, again, voltage gated. So because of this, calcium is going to enter into the axon terminal and causes these synaptic vesicles to fuse to the end of the axon terminal. In the fusion, acetylcholine gets spit out because of exocytosis and enters the synaptic cleft. And then acetylcholine is going to diffuse across the synaptic cleft and bind to an acetylcholine receptor. Okay, and so just for all intents and purposes, so you guys do get an appreciation of where we're at here. I'm going to just zoom, give you like a zoomed in picture. Drawing, I guess, rather. Okay, and so these little green dots is acetylcholine diffusing across the synaptic cleft, and the acetylcholine will bind to its receptor. Okay. Which is what you see here and here. And then when acetylcholine has bound to its receptor, this is going to cause the receptor to change its shape and open up the gates. And the opening of the gates is going to cause sodium molecules to rush into the muscle fiber, and potassium molecules will be rushing out of the muscle fiber. This is going to result in an in-plate potential because more sodium ions are entering than potassium ions leaving. Okay, so if you have, let's say for example, we have sodium coming in and we bring 3 sodiums in. Each of them has a positive charge. So now we brought in 3 positive charges, but say when potassium left, there was only 2 potassiums that left. If only 2 potassiums leave, then we only got rid of 2 charges, minus 2 charges. Therefore, we're left with now the inside having a +1 charge. Okay, so hopefully you understand how that works. If more positive comes in than is leaving, then that's going to lead to the accumulation of positive charges inside, right? And this is then resulting in what we call an in-plate potential. Now, finally, what has to happen is at the end of all of this, acetylcholine needs to be released from our nice little channel here, nice little ion channel. And so in order for that to happen, we have an enzyme called acetylcholinesterase that's going to come in and basically pluck acetylcholine off of this receptor, break it down, and recycle part of the acetylcholine. And then the rest of the acetylcholine, say we have more acetylcholine just hanging out here in the synaptic cleft, essentially what's going to happen is that acetylcholine is just going to diffuse away. All right, so here I'm going to share a video with you kind of animating this.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
So I think that's a really great video. I think it gives a really good animated vision to help you understand exactly what's going on here at the neuromuscular junction. So I want to just take a second to bring up the fact that this is relevant clinically. And we're going to talk about something called myasthenia gravis. So there's a lot of different toxins and drugs and diseases that can interfere with the events that are occurring here at the neuromuscular junction. And the example, again, that I'm going to talk to you about is myasthenia gravis. This disease is characterized by drooping upper eyelids, difficulty swallowing and talking, and general muscle weakness. But essentially, the pathophys that's occurring here is that there's a shortage of acetylcholine receptors because the receptors get attacked by the person's own antibodies. This suggests that myasthenia gravis is an autoimmune disease. And so here is kind of a quick picture of what normal muscle contraction, the neuromuscular junction would look like. But in myasthenia gravis, some of these receptors are being blocked by antibodies. And so acetylcholine can't actually bind to all of the receptors that it's supposed to be able to. Due to this, you will have a reduced muscle contraction. Okay, So now that we've covered the neuromuscular junction, we're going to move on and talk about how muscle fiber excitation works. So our resting sarcolemma is polarized, and polarized means that there's a voltage that exists across the membrane. And for our purposes, the voltage that's existing is we have a cell here. The inside of our muscle fiber, our muscle cell, is negative compared to the outside. Okay, so the inside of our cell is more negative than the outside of our cell. And action potential is going to be caused by changes in these electrical charges and occurs in 3 general steps. So we have the generation of the end plate potential, depolarization, and repolarization. So let's talk about each of these. The generation of the end plate potential is what we're first going to talk about. So acetylcholine that's released from our motor neurons are going to bind to the acetylcholine receptors on the sarcolemma, which we understand. And this causes the chemically gated ion channels on the sarcolemma to open. Okay. So sodium is going to come into the muscle fiber, and potassium is going to diffuse out. But not as much potassium is diffusing out as sodium is coming in. And because sodium is coming in, the inside of the sarcolemma is going to become less negative. So remember that I said for our cells here, our muscle cells, we had a negative inside of the cell. If we have positive charge coming in, we're basically canceling out some of these negative charges, right? Because a plus and a minus is a neutral, so we can cancel those out. And now we are less negative, okay, or more positive. All of this is going to result in a local depolarization, what we call a end plate potential. All right, so what happens next? What happens next is depolarization, and depolarization refers to the generation and the propagation of an action potential. So if the end plate potential, the one that we just talked about on the last slide, causes enough change in that membrane voltage, okay, so talking about depolarization here, if the end plate potential that we just talked about caused enough change to reach a critical level, which we call threshold, then voltage gated sodium channels in the membrane will start opening. And it's going to cause a huge influx of sodium through channels into the cell, which will then can trigger the action potential that is at this point unstoppable and will lead to muscle fiber contraction. And this action potential is going to spread across the sarcolemma from one voltage gated sodium channel to the next one in the areas that are surrounding, causing that area to depolarize. And this cycle is just going to continue on and on. And so just to kind of give you a quick little picture of a more expanded view here, I just quickly drew up this little example. So over here on the left side, this is the neuromuscular junction. And what's already happened now is the acetylcholine bound to its receptors on the sarcolemma. And if we remember, this causes sodium to rush in, and as sodium is rushing in, we have some potassium rushing out, but there's more sodium coming in. And this is going to cause this membrane here to now, instead of being negative, be positive. If enough change happens in this location, if there's enough change in this membrane potential, then this will stimulate our voltage gated sodium channels. Here, this pink channel to the right, it's going to stimulate that channel, and what's going to happen is that channel is now going to open. And when that channel opens, sodium, which carries a positive charge, is going to start flooding in to this area of the cell. This causes the membrane potential in this area to start becoming positive. As we move sort of down the line, essentially what would be happening is we would have more voltage gated sodium channels down the line. And because there was so much positive change here in this area, that stimulates this voltage gated sodium channel to open. And now sodium ions can start flooding in this section of the fiber, of the muscle fiber. Okay, and now that means the membrane potential in this section of the muscle fiber is going to start becoming positive. And this same process keeps proceeding the entire length of the muscle fiber. Okay, so hopefully that gives you a better picture as to what exactly is going on, and we'll talk a little bit more about it as we proceed through the lecture. So after the area of the membrane has been depolarized, it then becomes repolarized. And this just means we're going to go back to our normal resting state. So those sodium channels that were opened are now going to close or be blocked again. So now no more sodium can come flooding in. And when the sodium channels are closed, the potassium channels are going to open. And the potassium channels, whenever they open, this causes potassium to leave the cell. And so you should get an appreciation that if a positive charge is now leaving the inside and going out, that means we're going to be left with the membrane returning to its negative state, its negative inside state, which is going to be its initial resting membrane voltage. In kind of talking about this, it's also important to understand that there's what's called a refractory period. And the refractory period occurs when the muscle fiber can't be stimulated for a specific amount of time until it gets back to its normal state. And these normal resting state conditions are restored by the sodium potassium pump, or sodium potassium ATPase, which we talked about back in the cells lecture. And so as we hopefully remember, the sodium that came into the cell is now going to be pumped back out, and the potassium that was pumped out of the cell up here in this situation is going to be pumped back into the cell. So let's just quickly, I'm going to just draw this for you guys so we have a good understanding of what exactly this sodium potassium pump is, right? So put the sodium potassium pump, I'm going to just draw part of it on here. Right, so whenever the action potential kind of strolled through, we had sodium rushing in, right? Rushed into the cell. Well, now we have a whole bunch of sodium down here, and we don't necessarily want a whole lot of sodium down there. And so what we're going to do then is, using our sodium potassium pump, we're going to send that sodium back out, specifically 3 sodiums back out. And we are going to bring back in potassium, but we're only going to bring in 2 potassium molecules. So if more positive charge is leaving than is coming back in, this is going to reset this area of the cell membrane back to its normal negative. Oops, sorry about that. Back to its normal negative state. Okay, so we're back to our normal negative state because the sodium potassium ATPase is restoring these values. Okay. Okay, so in this next section, we will talk about and explain the concept of excitation contraction coupling. Okay, so excitation contraction coupling, or ECC, just describes the events that transmit that action potential along the sarcolemma, which is the excitation portion of ECC. And the excitation contraction coupling basically just combines the 2 ideas that we were just talking about, basically combining the idea that the sliding filament model is occurring at the same time as when our cells are being excited. So the action potential is going to be propagated along the sarcolemma and down the T-tubules. And here, this is where voltage-gated proteins in the tubules are going to stimulate the release of calcium. And calcium is what is actually leading to the contraction. Okay, So remember on the last several slides, we're talking about how the action potential gets propagated down the length of our cells, right? And when that action potential reaches a T-tubule, that T-tubule in and of itself essentially just diverts the action potential and allows it to dive kind of more deeply. And when that electrical signal reaches the sarcoplasmic reticulum, this is going to cause voltage-gated calcium channels to open. This is going to cause calcium to be released from this sarcoplasmic reticulum, thus stimulating the sliding filament model. This action potential, it's going to be very brief, and it actually is going to be ending before the contraction is even seen. Okay, so action potential arrives first and then contraction happens, which is why we call it excitation-contraction coupling. So again, it's the sequence of events by which transmission of an action potential along the sarcolemma leads to the sliding of the myofilaments. And so the action potential, so up here is just a zoomed in picture of essentially what I just drew on for you. The action potential arrives and propagates down the sarcolemma and the T-tubules. This stimulates calcium channels that are voltage sensitive to change their shape and release calcium into the sarcoplasmic reticulum. And thus into the cytosol. And then when calcium is released, it goes and it finds the myosin binding sites. And this thus is going to stimulate the cross-bridge cycle, which we will talk about momentarily. All right, so let's talk about, I'm going to show you a quick video on excitation-contraction coupling.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so now that we've covered all of that, we're going to move on and talk about the cross-bridge cycle, which I've brought up several times. And so let's learn about it. So cross-bridging is just referring to the different events that are happening when the myosin head is pulling those thin filaments towards the center of the sarcomere. And this process is dependent on the intracellular calcium concentration, which is essentially what we just talked about on that last video and the last couple of slides. And by intracellular calcium concentration, I'm meaning how much calcium is inside of the muscle fiber. Okay, so at low calcium levels, if there's not very much calcium inside the cell, it's sort of stored away at the time, then tropomyosin is actually blocking the active sites on actin. And therefore, because of this, the myosin head is blocked and it can't attach to actin. If this is the case, then the muscle is going to remain relaxed. When an action potential arrives and the voltage sensitive proteins in those T tubules change their shape, and that causes the sarcoplasmic reticulum to release calcium into the cytosol of the cell, now we have an increasing or a high intracellular calcium concentration. Okay, so now we have a lot of calcium over here. Calcium is going to bind to troponin, which are the little yellow structures that you see here. So the little yellow structures, calcium binds to troponin. Troponin causes tropomyosin, which is those orange lines, to shift away from the active sites that are on actin. Because of this, then now you can see that the myosin binding sites on actin, those little divots, are now available for the myosin head to latch onto. This begins the cycle, and this causes a sarcomere shortening and muscle contraction. Now, whenever the nerve stimulation stops, then this calcium, all this calcium gets pumped back into the sarcoplasmic reticulum, and the contraction is going to end. So cross-bridge cycling occurs in 4 steps. Step 1 is the cross-bridge formation. So the cross-bridge formation occurs when the myosin head attaches to the actin thin filaments on their active sites. So step 1, myosin latches onto actin. Step 2 is the working or the power stroke. So the myosin head in the power stroke is going to pivot, and it's going to pull the thin filament towards the M line. Okay, so we started sort of up in this position, and now we're sliding the actin filament to the left. ADP and inorganic phosphate are going to be released, and that's what causes the power stroke to actually occur. So originally we have ADP and inorganic phosphate attached to the myosin head during the cross-bridge formation, and in order for the myosin to actually slide the actin, we have to release this ADP and inorganic phosphate. The next step of the cross-bridge cycle is that the myosin head is going to detach. And this occurs after ATP, or energy, attaches onto the myosin head. So we just got rid of ADP, and now ATP has to come in and attach to the myosin head, which will cause the myosin head to pull away from actin. And then the 4th step here is now we have to move this head back to its cocked position and get ready to latch on and slide the actin filament again. And essentially we do that by breaking down ATP into ADP and inorganic phosphate. All right, and this causes the myosin head to return to its pre-stroke, really high energy cocked position, and then the cycle continues. Okay. So let's see that in action.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
So I always think it's easier to understand things when you can see them in sort of a animated way. So I love putting in these videos whenever I can, and I hope they do really help you. So before I finish up part 2 here, I just want to make a clinical connection here to rigor mortis. Rigor mortis is the medical term that we use to describe the stiffening of body muscles that occurs after death. And it begins about 3 to 4 hours after death. And peak rigidity, so when the muscles become most rigid, is usually about 12 hours post-mortem. So the reason that this is occurring is because the intracellular calcium levels are going to increase because ATP is no longer being made. Once a person dies, um, obviously the energy production would stop. So since ATP is no longer made, the calcium can't be pumped back into the sarcoplasmic reticulum. And this results in cross bridge formation, right? Because if calcium is present, then cross bridges are going to be formed. But remember, ATP is also needed in order to detach those myosin heads. So if we are not making ATP, then the myosin head is going to stay bound to actin, and this is going to cause a constant state of muscle contraction. The muscles are going to stay contracted until the muscle proteins themselves actually break down during decay, and that's what's going to cause the myosin to be released. And so just a kind of an interesting clinical connection that we can make to what we're learning about is, I think, always a fun way to integrate our learning. All right, so I am going to end part 2 video here, and I will come back for part 3 where we'll cover the rest of this information.